Hi, good night, everyone. My name is David Amra. I'm one of the Part 1 surgery residents in orthopedic surgery. Today, I'll be presenting and talking on the endocrine functions of the pancreas and the liver. So, the objectives for tonight's presentation will be to define the endocrine versus exocrine function and to highlight the differences between both of them. To highlight the main endocrine hormones secreted by the pancreas and the cells which secrete each hormone, to highlight briefly the physiologic effects of each of the hormones listed, and as well to describe the mechanisms involved in secretion of the various pancreatic and endocrine hormones, and to highlight the endocrine functions of the liver. So, brief anatomy overview. The pancreas is a retroperitoneal organ, so roughly 12 to 15 cm in length. It's a horizontal organ which is positioned behind the stomach, and the head is the widest part, and it rests within the C loop of the second part of the duodenum. Then you have the uncinate process, which is a hook-like extension, which loops around, projecting inferiorly and posteriorly from the head, as well as the neck, and then the neck, which is the short segment connecting the head to the main body, and then the body, which is the main section stretching horizontally behind the stomach in front of the splenic artery, and then the tail, which is tapered to the left, touching the spleen, the splenic hilum. So there you could just see a very small diagram. This is not an anatomy discussion, but just a small diagram to understand the anatomy and understand and remember the anatomy of the pancreas. Now, the gross anatomy of the pancreas is more important for the exocrine secretion. However, understanding that the venous supply, the venous drainage, of the pancreas drains into the portal venous system is of paramount importance to understand the endocrine function of the pancreas. So the core structural features are the main pancreatic duct would traverse the entire gland horizontally and open into the armpit or vata. And you have your two ducts, the duct of Wurtzbach, which is the main duct, and the duct of Santorini, which is the accessory duct. And essentially the common bile duct would merge in the main pancreatic, in the inside pancreatic head to form the common bile duct. Now, moving on to the real meat of the discussion tonight, which is the endocrine. So the endocrine versus the exocrine. So exocrine is basically where the hormones are secreted directly through ducts. And the endocrine are more ductless hormone secretions that are secreted into the bloodstream. So one is secreted onto a more epithelialized surface, whereas the other one is secreted directly into the blood. So you have local secretion, which is the exocrine secretion, and it would act on nearby cells or tissues. It will drain directly into the gut with respect to the pancreas, whereas in the endocrine pancreas, the hormones are secreted more into the bloodstream, which feeds into the portal venous system, which we'll touch on later. The exocrine would consist more of enzymes, which are like large protein-like molecules, whereas the endocrine would be more consisting of hormones. The exocrine would have local action, the endocrine would have distant, widespread action throughout the body, and the exocrine, some examples would be, for example, the salivary glands, sweat glands, sebaceous glands. Some examples of the endocrine glands would be the pituitary gland, the thyroid gland, the adrenal gland, the gonadal system, and tonight we will hear about the pancreas and the liver. So the endocrine hormones of the pancreas were essentially discovered in 1869 by a gentleman named Paul Langerhans, who discovered the islets of Langerhans, which were discovered to be the functional unit of the endocrine pancreas. These islets were found to be tiny clusters of cells that synthesized hormones within the pancreas. A very small portion of the weight of the pancreas are the islets of Langerhans. They appear as pale oval islands under a microscope, and they are small collections of cells. Now, a very important aspect of the islets of Langerhans is that they have a very vast blood supply, and they're packed with tiny fenestrated capillaries for fast dispersal of the endocrine hormones into the bloodstream. And it's the only endocrine organ that drains directly into the hepatic portal vein. So here's a small slide of the islets of Langerhans and a more well-depicted diagram on the right, which highlights the various cells of the islet of Langerhans. So the beta cells, the alpha cells, the delta cells, as well as the pancreatic acinar cells. There are two other cells which are not depicted in this diagram here, which would be the delta cells, sorry, not the delta cells, the gamma cells, as well as the epsilon cells, which secrete smaller amounts of hormones. So here's just a more in-depth picture of the islets of Langerhans to understand that the capillaries that perfuse the islets of Langerhans are key in terms of the taking away the hormones to feed into the bloodstream. All right, so key features of the islets of Langerhans, they're only encapsulated by a one-cell thick cell wall. They have a rich sympathetic and parasympathetic nerve supply. They are highly vascular, even though they're only, as I said previously, 1 to 2% of the mass of the pancreas. They receive a very large 15% of the total blood flow of the pancreas. And the blood flow drains straight into the portal vein and will take part in a system known as the insular acinar portal system to exert effects on the exocrine pancreas. So the capillaries of the islets are fenestrated, which will allow the hormones to perfuse readily into the blood supply to feed into the insular acinar portal system. And the cells are organized well positioned around the capillaries. So moving on to the contents of the islets of Langerhans and the hormones that are produced. So the most important one for the talk tonight would be the beta cells, which are 60 to 80% of the islets of Langerhans. These cells produce insulin and another very small amount of a hormone called amylin. Then you have your alpha cells, which are 15 to 20%, and they produce glucagon. You have your delta cells, which are 3 to 10%, they produce somatostatin. You have your gamma cells, which produce pancreatic polypeptide, and your epsilon cells, which are less than 1%, that produce the very important hormone ghrelin. So moving on to insulin, up in the corner here, we see the structure of insulin, which are two chains of large amino acids linked by disulfide bridges. So it's a 51 amino acid polypeptide containing two chains of amino acids linked by disulfide bridges. So essentially, it is a hormone of abundance, and it is synthesized in the rough endoplasmic reticulum of the beta cells as a structure called pre-proinsulin, which is coded for the gene of the short arm of chromosome 11. So pre-proinsulin is then cleaved to form proinsulin, which is transported to the Golgi apparatus, where further cleaving and packaging occur into granules with C-peptide. These granules are released by exocytosis on stimulation of the beta cells. So the pancreas contains, at rest, 200 units of insulin stored in the beta cell granules, and it secretes roughly 0.25 to 1.5 units per hour under normal conditions. So here's a small diagram about insulin formation. So it shows where it's formed in the beta cells, down to proinsulin, where it's packaged in the ribosomes and stored in the Golgi apparatus, and where there's proteolytic cleavage to break it down into insulin and C-peptide, and it enters the portal circulation where it exerts its effects. So moving on to the insulin effects. The target effects take place through the insulin receptor. How the insulin works, it First, it helps, what it does, it uptakes glucose into the cells. It's important in the uptake and storage of carbohydrate as glycogen. And it does this through the stimulation of glycogenesis, which is creating glycogen, and amino acid uptake, and anabolic protein synthesis, as well as fats and conversion of excess glucose into fatty acids. So essentially, it packs away all the substrates of the body, the major substrates. So it inhibits gluconeogenesis, so it inhibits the formation of glucose, as well as it promotes glycogen. It does not, sorry, it promotes glycogenolysis. Sorry, it inhibits gluconeogenesis and glycogenolysis. And it acts on adipose tissue to increase the size of the adipose tissue, as well as skeletal muscle and liver by packing away through glycogenesis. So there's a small diagram on the effects of insulin. So the main effects would take place in the liver, where it decreases glucose synthesis, increases glycogen synthesis, increases lipid accumulation, increases inflammation. In the adipose tissue, it increases glucose metabolism, increases lipogenesis, decreases lipolysis. And in the peripheral muscle, it would increase glucose metabolism, increase glycogen synthesis, and increase mitochondrial dysfunction, and increase muscle mass. In the brain, it would increase hunger, decrease hepatic glucose production, and decrease lipoprotein production. So this is a nice table which highlights the effects of insulin. So in the immediate secretion phase, it would increase the transport of glucose, amino acids, potassium into the insulin-sensitive cells. And within minutes, it would start to stimulate protein synthesis. It would inhibit protein degradation. It would activate glycolytic enzymes and glycogen synthase. It would inhibit phosphorylase and gluconeogenic enzymes. Within hours, it would increase the mRNAs for lipogenic and other enzymes. So here are some tissue-specific activities of insulin. So the adipose tissue, it increases glucose entry, increases fatty acid synthesis, increases glycerol phosphate synthesis, increased triglyceride deposition, increased lipoprotein lipase, and inhibits hormone-sensitive lipase, and increased potassium uptake. So essentially, it packs away substrates within the storage centers of the body. In the muscle, basically the same thing, more or less, with glucose entry, glycogen synthesis, and increased amino acid uptake, increased protein synthesis, decreased catabolism, as well as increased ketone uptake and increased potassium uptake as well. Within the liver, there's decreased production of ketone bodies, as well as there's increased protein synthesis, increased lipid synthesis, and decreased glucose output due to decreased gluconeogenesis, increased glycogen synthesis, and increased glycolysis. And in general, there's increased cell growth. So this is how insulin exerts its effects through the insulin receptor. What is the insulin receptor? It is a four subunit receptor consisting of 2 alpha units outside the cell and 2 beta units that penetrate the cell membrane. The insulin binds to the alpha subunits on the outside of the cell, and autophosphorylation activates tyrosine kinase. It phosphorylates other tyrosine kinase, then goes on to phosphorylate other intracellular enzymes known as insulin receptor substrates. So through the insulin receptor substrates, IRS1 and IRS2, IRS3, insulin directs the intracellular metabolic machinery to produce the end desired effects on carbohydrate, protein, and fat metabolism. So this is a small diagram depicting the effects of insulin on the insulin receptor. Here you can see the alpha units on the outside of the cell, the beta units going on the inside of the cell, where they activate the tyrosine kinase and the insulin receptor substrates. And you see their various effects on fat synthesis, protein synthesis, glycogen synthesis, growth and gene expression, and ultimately moving the GLUT4 receptors to the cell membrane as well, which we'll touch on later. So insulin effects on GLUT4. So insulin stimulates the uptake of glucose into adipocytes, cardiac muscle cells, skeletal muscle cells, as well as the hepatocytes via the GLUT4 transporter. So the key steps would be molecular activation. So as we discussed just now, it binds to the extracellular alpha subunit of the insulin receptor. The activated receptor recruits and phosphorylates the IRS proteins. The IRS proteins bind and activate the phosphorylated inositase enzymes, which convert the other phosphoinatidyl enzymes to the last one, which is PIP3. And there's accumulation of the PIP3, which would activate downstream master kinases to primarily activate the AKT protein kinase B. So the AKT inactivates AS160, which normally keeps the GLUT4 locked inside the vesicles. And it switches a protein called the Ra protein to the active GTP bound state. And then the vesicles containing the GLUT4 detach from the cytoskeleton and travel to the membrane, the cell surface membrane, where they fuse with the plasma and have the GLUT4s now located on the membrane, where they're exposed to the extracellular fluid and aid in glucose entry permitted down its concentrated gradient. Here's a nice diagram just to understand the actions of the GLUT4 receptors as they're brought out onto the cell membrane to bring the glucose within the cell. So here you can see, as we spoke about, the cascade with the insulin receptor, the IRS cascade, as well as straight down to the kinase mechanism, which activates the GLUT4 vesicle, which translocates the GLUT4 receptors straight to the cell membrane to activate and bring in the glucose into the cell. So in terms of the physiology of the insulin secretion, the insulin secretion from the human pancreas is independent of it's dependent of the metabolic and neurohormonal stimuli, which occurs every 6 minutes. So without any metabolic or neurohormonal stimuli, it occurs independently every 6 to 8 minutes. And the most important stimuli for active release would be glucose. So once the serum glucose rises into the beta cell via a GLUT2 receptor, via facilitated diffusion, it goes inside of the cell and undergoes metabolism to form ATP in the beta cell. The ATP would inhibit the KATP sensitive channels, and this would depolarize the cell and increase the calcium stimulating insulin release by exocytosis. So the response to glucose is biphasic. You have a rapid, short-lived increase of insulin secretion, followed by a slowly developing prolonged increase. The fasting normal blood level of insulin is 0 to 7 micromoles per milliliter, released at a rate of 1 unit per hour, and it's increased by 5 to 10-fold following a carbohydrate-rich meal. So the biphasic secretion explained. So initially it would enter via the GLUT2 transporters, and it's phosphorylated by glucokinase, which is then metabolized to pyruvate. Pyruvate via the citric acid cycle produces ATP, which enters the cytoplasm of the beta cell. ATP sensitive channels are inhibited by the beta cell and and the beta cell is depolarized when calcium enters. And calcium causes the exocytosis of the readily releasable pool of insulin granules via exocytosis. So the metabolism of pyruvate via the citric acid cycle would then cause the increase in glutamate, which would act on a second pool, which would release these granules in a prolonged second stage. So here you just see the action in the beta cell via the GLUT2 receptor, which allows the glucose into the cell via facilitated diffusion, where it undergoes a cascade into the citric acid cycle with ATP and affects the potassium out of the cell, depolarizing the cell. And what it does is it causes the vesicles to move to the cell membrane and secrete the insulin. So factors affecting insulin secretion. You have your stimulators of the insulin secretion and the inhibitors of the insulin secretion. So stimulators, the biggest one is glucose. Then you have mannose, amino acids, certain intestinal hormones, keto acids, acetylcholine, glucagon, cyclic MP, as well as beta-adrenergic stimulants, theophylline, sulfonylureas. The inhibitors, you have your somatostatin, you have deoxyglucose, hepatulose, alpha-adrenergic stimulators, so norepinephrine, epinephrine. You have your beta-adrenergic blockers. You have different things such as thiazide diuretics, potassium depletion, phenytoin, as well as insulin. So now the other small hormone, which is not that, I wouldn't say it's not important, but the amylin, which is secreted as well from the beta cells. It's a 37 amino acid polypeptide, which is co-secreted with insulin by the beta cells, and it's more or less a glucoregulatory hormone. And it's secreted by the Golgi apparatus as an 87 residue pre-hormone, and it's secreted directly along with insulin in a molecular ratio of 100 insulin to 1 amylin. And what it does is it slows the rate of nutrient and glucose entry into the bloodstream by decreasing the rate of gastric emptying. So essentially it mitigates the heavy effects of glucose. And it inhibits the postprandial glucagon release, and it acts directly centrally on the medulla oblongata postrema to increase feelings of satiety and fullness. Moving on to the alpha cells of the pancreas, you have the glucagon secreted by these cells. It's a large 29 amino acid polypeptide with actions directly opposed to insulin. So you have 0.7 to 0.8 milligrams stored in the adult pancreas, and the major effects on glucose metabolism include a breakdown of liver glycogen, glycogenolysis, and increased gluconeogenesis in the liver. And it increases the availability of glucose throughout the body, and it can be termed the hyperglycemic hormone. So the physiological effects of glucagon. How does it cause glycogenolysis? Basically, it activates the adenyl cyclase in the hepatic cell membrane and causes the formation of ANP. And then the protein kinase activation and conversion of phosphorylase B to phosphorylase A will promote the degradation of glycogen to glucose 1-phosphate, which is then dephosphorylated and released from the liver cells. So this is a diagram to understand the relationship between the concentration of glucagon in the bloodstream and with respect to the blood glucose concentration. So high glucagon in low blood glucose and low glucagon in high blood glucose. So the actions again, glycogenolytic, gluconeogenic, lipolytic, ketogenic, but it does not cause glycogenolysis in muscle. And large doses have been shown to have a positive inotropic effect on the heart without producing increased myocardial excitability. Here's a small diagram to understand the effects of glucagon on the body, throughout the body. So you see the various effects. In brown tissue, the breakdown in brown adipose tissue, the increased insulin secretion in the pancreas, increased insulin synthesis. You see the brain, decreased food intake, decreased body weight. You see the liver, increased gluconeogenesis, increased glycogenolysis, as well as break down all the substrates which can be stored within the body. The GI tract, it decreases gastric emptying, decreases GI motility. The heart, it increases the heart rate, increases cardiomyocyte survival, as well as in white adipose tissue, it releases the ketone bodies into the blood circulation. So factors which affect glucagon secretion, you have amino acids which stimulate, glucose which inhibits, gastrin and cholecystokinin which stimulate, somatostatin which inhibits. You have cortisol which would stimulate, secretin which would inhibit. You have exercise which would stimulate, and free fatty acids which would inhibit. Other things such as infections, stresses, beta-adrenergic stimulators, theophylline, acetylcholine would stimulate the production of glucagon, whereas things such as insulin, phenytoin, and alpha-adrenergic stimulators and GABA have been shown to inhibit glucagon secretion. A nice diagram which will help understand the differences between insulin and glucagon. So basically, glucagon raises back up the glucose from a low level, and the insulin drops the glucose from a high level. So here as well, it goes into the function of the glucagon on the cell membrane through the adenyl cyclase mechanism, which activates protein kinase down with the phosphorylation downstream enzymes, which goes on to the catabolic processes, which break down glycogen and lipids and ultimately lead to gluconeogenesis, as well as the inhibition of the anabolic processes such as glycogen synthesis, protein synthesis, and lipid synthesis. It's a nice diagram which highlights all the effects of glucagon. Moving on to the delta cells now, the somatostatin. So it's a regulatory peptide hormone which is secreted from the delta cells of the pancreas. And it acts via a paracrine method to exert its effects on the pancreatic hormone secretion. So essentially, it's secreted in two forms, SS14 and SS28, and it's synthesized as pre-prosomatostatin, mainly by the gut and the brain, but 5% of it is secreted by the pancreas. And it's stimulated by lipids, carbohydrates, amino acid, luminal contents, gastric acid secretion, cholecystokinin, gastrin, and secretin. It's suppressed by fasting, muscarinic, cholinergic agents, and adrenergic antagonists. Some of the effects of somatostatin: it inhibits the secretion of pancreatic and pituitary hormones, inhibits exocrine and endocrine secretions of the pancreas, inhibits intestinal secretions, including the gastric secretions, inhibits gallbladder contraction, inhibits splenic blood flow, and upper GI motility. We know its analog used readily in medical settings, known as octreotide. So the pancreatic polypeptide is secreted by the PP gamma cells, and essentially it's a 36 amino polypeptide, which plays a regulatory role in digestion and metabolism. It would inhibit the pancreatic endocrine and exocrine secretions, inhibits gallbladder contractions, decreases small intestine motility, and would increase colonic motility. It inhibits appetite, increases hepatic insulin sensitivity, and increases glucose uptake into hepatocytes. It is stimulated by gut luminal contents, and protein being the most stimulatory of the pancreatic polypeptide secretion from the gamma cells. Last but not least, the epsilon cells produce a hormone called ghrelin, which is shown to be released before each meal. It drops approximately 1 hour postprandially, and very little is secreted from the pancreas. The effects include increased growth hormone secretion, increased stomach acid, increased GI motility, and increased appetite. Moving on now to the second part of the talk tonight, the endocrine functions of the liver. So the liver functions it functions in different ways to exert endocrine effects within the body. So the liver functions to produce key hormones. It functions to modify and break down other hormones. And it also functions to produce hepatokines, which are specialized liver proteins that function as hormones to control various bodily functions. So the direct hormone production from the liver, you have IGF-1, which is secreted in response to growth hormone to promote general cell growth and manage how the body uses nutrients. Then you have angiotensinogen, which is an essential alpha-2 globulin glycoprotein produced primarily by the liver that serves as a precursor for all angiotensin peptides, which is a key component of the renin-angiotensin-aldosterone pathway, which plays a major role in blood pressure homeostasis. As well, fluid volume and electrolyte balance in the body. And then you have the 25-hydroxyvitamin D, which is a pro-hormone that is later converted to active vitamin D, which is very important in the calcium homeostasis within the body. Then you have the last hormone, which is hepcidin, which is a regulator of human iron metabolism, and it controls iron absorption, recycling, and storage. So the most important one would be the hepcidin. which would be a peptide hormone produced by the liver. It would play a role in iron homeostasis. It decreases iron levels by reducing dietary iron absorption and inhibiting iron release from cellular storage. The production increases when iron levels rise above 65 to 175 micrograms per deciliter in males, and when it rises above 58 to 178 micrograms per deciliter in females. It acts through ferroportin 1, an iron export transmembrane protein, which is present in macrophages and erythrocytes. So the hepcidin attaches to ferroportin 1, causing degradation, which blocks the absorption from enterocytes of the gut and release of iron from the macrophages, which recycle iron from degraded erythrocytes in the bone marrow, liver, and spleen. So in terms of hormone metabolism and clearance, the liver plays an important role in systemic hormone concentration balance by directly breaking down or modifying hormones for excretion, as well as managing cholesterol, which is the building block for steroid hormones. So steroid hormones are inactivated and conjugated by the liver. So the various hormones such as cortisol, aldosterone, estrogen, and testosterone are both inactivated and conjugated by activities of the liver. As well, the liver converts T4 to T3, which is a central thyroid hormone, which is more active in the T3 state. In terms of the synthesis of the binding proteins, the liver plays a crucial role in the production of carrier proteins, so binding globulins, which can determine the amount of free available hormone. So in terms of the hormones which the liver controls via the production of carrier proteins, you have the sex hormones, so testosterone and estradiol, which the concentrations may be altered by the levels of sex hormone binding globulin, as well as the thyroxine binding globulin, which would carry T3 and T4. As well, you have your corticosteroid binding globulin, which is known as transcortin, which would bind the cortisol and aldosterone. So the liver takes up amino acids from the bloodstream. It assembles polypeptide chains through DNA-coded mRNA onto the ribosomes of the rough endoplasmic reticulum of hepatocytes and modifies them in the Golgi apparatus and secretes the functional binding globulins into the bloodstream. So here's a nice table which would just briefly go through the endocrine functions of the liver. So from the vitamin D production, so the 25-hydroxyvitamin D, the liver directly produces it. It has its target activities on the gut. And what it does is a prohormone of calcitriol, which stimulates calcium absorption from the gut. And essentially, this table also highlights what happens in liver pathology. So there's a decreased production resulting in low bone density when the liver is affected. So that's how you see that the liver does have a very important role in production of 25-hydroxyvitamin D. As well as the insulin-like growth factor, the liver again directly produces this hormone. And what this hormone does is it has widespread effects throughout the body, which promotes growth and differentiation through regulation of nutrients and metabolism. Decreased production would result in dysmetabolism. Again, another direct production hormone, angiotensinogen. You see its target effects on the cardiovascular system. It is a precursor of angiotensin 2, which regulates aldosterone levels, which both regulate the vascular tone, sodium retention, and cardiac remodeling. Now, in liver disease, you have near normal function, so it's not that important from the liver. Then you have your thyroid hormone, which would have your the liver, what it does is it activates it through T4 to T3 conversion, and it also inactivates it through degradation, and it also has effects through the thyroid globulin production. This has widespread effects throughout the body by increasing metabolism and energy expenditure. When the liver is damaged or there's liver pathology, there's low T3 syndrome. You have some other important hormones here, so GLP-1. So the liver functions to metabolize GLP-1 via the dipeptidyl peptidase DPPIV. And what it does is it has target effects on the pancreas, gut, and brain, and it stimulates insulin production, decreases gut motility, and suppresses the appetite. So when the liver is damaged, there's increased DPPIV expression, resulting in a higher risk of diabetes, as well as the sex hormones are secreted, which have direct effects throughout the body on the liver functions in terms of the metabolizing the hormones, breaking them down, as well as producing the carrier proteins discussed previously. When the liver is damaged, we see hypogonadism, and the actions are numerous throughout the body through secondary sexual characteristics development and maintenance. Then you have glucocorticoids, which the liver functions to metabolize the hormones, as well as have the carrier proteins we spoke about previously. The glucocorticoids have widespread effects throughout the body, and the effects of the glucocorticoids are very widespread throughout the body. And when there's alteration in liver disease, there is relative adrenal insufficiency. As well, lastly, you have the mineralocorticoids, and the liver functions in terms of metabolizing and breaking down the hormones. It has effects on the cardiovascular system, and the mineralocorticoids function to maintain the electrolyte balance and the blood pressure. So moving on, the other endocrine function of the liver would be secretion of some hormones known as hepatokines. So hepatokines are secreted exclusively or predominantly by the liver, and they act as hormones to regulate systemic metabolism. These hepatokines travel through the bloodstream and communicate with tissues like skeletal muscle, adipose tissue, and the pancreas. So some of the physiological functions: energy homeostasis, metabolic control, and exercise adaptations. Stimuli is controlled by nutritional states, fasted versus fed, physical exercise, endocrine and pancreatic hormones, glucocorticoids, and thyroid hormones. It's implicated in the development of non-alcoholic fatty liver disease, obesity, and metabolic syndrome. So some of the major hepatokines and roles. So you have your angiopoietin-like proteins, which are a group of secreted lipoproteins, and they are regulators of lipid metabolism. So you have at least 4 of them listed here: ANGPTL3, ANGPTL4, angiopoietin-like protein 6 and 8. Then you have another one named Phytuin A, which is a glycoprotein which will inhibit the tyrosine receptor kinase A and would modulate insulin sensitivity and pathways. Then you have fibroblast growth factor 21, which would decrease plasma glucose and lipid levels, increase insulin and leptin sensitivity, and decrease hepatic steatosis. Then you have follistatin, which is a metabolic regulator. You have GDF15, which would be increased in inflammatory states. You have hepasocin, which would regulate hepatocyte proliferation and metabolism. You have insulin-like growth factor, which is very important, but it functions like insulin and it promotes hypertrophy of skeletal muscle. You have LECT2, lipocalin-13, which is an insulin sensitizer, selenoproteins P, SMOC1, and the last one found on this article was TUSHKI, which would regulate energy expenditure, cholesterol balance, and metabolic stress responses. So these are some of my references: Ganogs, Guyton and Hall. So the articles, so the hepatokines were discovered in this World Journal of Hepatology, and some more stuff with the AMLN. The main bulk of the presentation came from the endocrine functions of the pancreas, which was a board exam for physiology online by Alex Yastev. Thank you.